Hello, everyone. During the previous lecture, I explained the basics of transcriptomics analysis and specifically of real-time PCR. And as we discussed, primer design is an important aspect related to qPCR. So that is what I will be explaining in more detail today. But before I start explaining which parameters are important to consider during primer design, I will first repeat some of the information from the previous lecture. Like a classical PCR reaction, also cycles of a qPCR reaction consist of three different steps. The first step is denaturation, which means that the two strands of double-stranded DNA will separate. And the second step, primers will anneal. So this means that they will bind to their complementary sequence. And subsequently, the primers will elongate in that way, again, forming double-stranded DNA. And through this process, the amount of PCR product is doubled with each PCR cycle. The difference between a classical PCR reaction and a real-time PCR reaction is that during real-time PCR, the generation of PCR product is monitored in real time, as the name implies. And this is done by using a DNA binding fluorescent probe. And the most important ones are CyberGreen, TOCMON, and molecular beacons and scorpion probes. In this lecture series, we will specifically focus on real-time PCR using CyberGreen. So I will briefly explain to you again the basic principle of CyberGreen probes. So both CyberGreen and also EvaGreen, which is a very similar dye, they will intercalate between double-stranded DNA. And when they're freely available in a solution, they will emit low fluorescence, but when they intercalate between double-stranded DNA, their fluorescence intensity will significantly increase. So this is what we can see here. So this is the first step of a PCR cycle, which is a denaturation. We have here in yellow our primers, and in green we have different CyberGreen molecules. But as you can see here, the two strands of the DNA are denatured, and CyberGreen is freely floating in the solution and is emitting only very low fluorescence. In the next step of the PCR cycle, the primers will anneal and subsequently they will extend. And as you can see, the CyberGreen molecules will intercalate between the two strands of the double-stranded DNA, and their fluorescence will strongly increase. This principle is exactly the same for EvaGreen, but this is a new dye with an improved performance as compared to CyberGreen. The advantages of using these kind of probes are that they're relatively simple. They also have a relatively low cost compared to certain other probes. And what's very nice is that they can be used to quantify the expression of any gene of interest. However, they also come with an important disadvantage, which is that they are nonspecific. So this means that these dyes will bind to any type of double-stranded DNA. So this means that they will also bind to primer dimers and other aspecific products. And this will, of course, also increase their fluorescence, hence making the obtained results unreliable. So this is, of course, something that should be avoided. Then to obtain data using real-time PCR, what the device will do is it will generate a so-called amplification curve for each sample. So you can see an example of such amplification curves in this graph over here. So on the x-axis, you can see the number of cycles that have passed during a PCR reaction. And on the y-axis, you can see the fluorescence intensity plotted in a logarithmic scale. And each curve here corresponds to the amplification in one specific sample. And then what the idea is behind real-time PCR is that we want to compare gene expression levels between different samples. And this comparison is based on the so-called CQ values. And CQ stands for quantification cycle. It's also called CT or threshold cycle. And this CQ or CT value corresponds to the number of cycles that are required for the fluorescence to reach a predefined threshold. This threshold is shown as this horizontal orange line here, and it's placed more or less in the middle of the exponential phase, or this exponential phase is also called a log linear phase, as it is a linear curve when the fluorescence is plotted on a logarithmic scale. And the CQ value can then be read here. So it's the number of cycles that a certain sample needs to reach this fluorescence threshold. And the CQ value is inversely proportional to the gene expression level. So this means that a sample with a low CQ value has a high expression of this specific gene that we're interested in, whereas a high CQ value indicates a low expression of the gene of interest. And the reason for this is that a sample with a high expression of the gene of interest will already have a higher number of mRNA copies, hence also a higher number of cDNA copies of this specific gene. And as a result, less amplification cycles are required to reach the same amount of DNA or the same amount of fluorescence. Then, after generating the amplification curves, the device will also generate a so-called dissociation curve or melting curve. This is done at the end of the qPCR run, and the main aim of this curve is to verify product specificity. Because as I mentioned, cybergreen will also be able to bind to aspecific products like, for example, primer dimers. How does the device do this? Well, it will gradually increase its internal temperature. And what will happen is that when the melting temperature of the specific product of interest is reached, the DNA will denature. As a result, cybergreen is released, and the fluorescence will significantly decrease. And this is what's plotted here on the so-called dissociation curve. So on the x-axis, we can see the increasing temperature. And on the y-axis, you can see the change in fluorescence per 1 degree increase in temperature. So this means that at the point where you see this peak, this will be the melting temperature of the specific amplicon. So for this upper sample here, this looks perfectly fine because we see one peak with melting temperature that's expected for the specific amplicon. However, in the sample that we see at the bottom here, we can see an additional peak. And this additional peak means that in addition to the specific amplicon, also an aspecific product is formed. In this case, we see that the second peak is more towards the left, which means that it's a product with a lower melting temperature. So it's very likely shorter product, and this will most likely be primer dimers. On the other hand, you could also see a second peak at a higher temperature, and this would indicate a longer product. And this could, for example, be genomic DNA, and I will come back later on why this is the case. So it's during qPCR, as I mentioned, an important step is to design primers. Of course, as we saw in the first slide of this lecture, primers are used to amplify specific part of our gene of interest. And as such, these primers need to be specifically designed for each gene of interest that we want to measure. And when doing so, there are different parameters that we need to take into account. And I've listed them here. So the first one is the amplicon length. And there is also the primer length, the primer melting temperature, self-complementarity of the primers, and also their binding location. And in the next slides, we will discuss each of these parameters in more detail. Starting with the amplicon length. So the amplicon is the PCR product that is being amplified in each cycle. And for qPCR, the ideal amplicon length ranges between 90 and 150 base pairs. So note that it's much smaller than a product that is generated during a classical PCR. So for classical PCR, you can amplify products of 1,000 base pairs or even larger. But for qPCR, we specifically amplify a relatively short product. Now, what would happen if we would try to generate an amplicon that is too long? Well, in this case, the amplification would take too long. And in a standard qPCR protocol, there would be insufficient time to fully amplify the entire product. Because in a classical qPCR protocol, the time foreseen for primer extension is only 15 seconds or even less. So if a product would be much longer than the 150 base pairs stated here, then there would simply be insufficient time to amplify this product. On the other hand, if the amplicon would be too short, this would be a problem as well. First of all, because this would make it very difficult to distinguish the specific amplicon from primer dimers in a dissociation curve. So here you can see again this example of a dissociation curve that we've just discussed. So here, this right peak corresponds to our specific amplicon. The peak more towards the left corresponds to primer dimers. These primer dimers have a lower melting temperature because they're shorter. But if we would now generate an amplicon that is also much shorter, then it would be very difficult to still distinguish it from primer dimers, as it would also have a lower melting temperature. And an additional issue when generating an amplicon that is too short is that less fluorescent dye would be able to bind. So you can see here an example of a piece of double-stranded DNA. And you see that there is space for 5 cyber green molecules to intercalate between the 2 strands. However, if the product would be shorter, less molecules would be able to intercalate. They would emit a lower fluorescent signal, and as a result, it would take longer. So it would take more cycles for the signal to increase above the fluorescence background. An additional parameter to take into account, in addition to amplicon length, is primer length. So the ideal length for primers for qPCR is between 18 and 25 base pairs. What would happen when primers are too long? Well, first of all, this would create a higher chance of formation of hairpin structures. So a hairpin structure is what is shown here on the right. And this means that a primer has sequences at both ends which are complementary to each other. So what would happen is the primer would sort of fall back onto itself so that both ends would be able to bind to each other. And this would cause the formation of this loop structure. In addition, primers that are too long also have a higher chance on forming primer dimers. The reason is that with longer primers, there is a higher likelihood that at least some part of the primers will be complementary to each other, and hence 2 primers might bind to each other. And another issue with primers that are too long is that their melting temperature would become too high. And melting temperature is something we will discuss in the next slide. On the other hand, when you would generate primers that are too short, this might also be a problem, because in that case, it would be very difficult to design primers that are specific only for your gene of interest. So let's say you design a primer that's only 5 bases long. So let's say A, G, T, C, G. In that case, it's very unlikely that this primer will be complementary only to a region inside your gene of interest, because very likely you will also be able to find complementary sequences at other locations inside the genome of your organism of interest. So with primers that are too short, it will be very difficult to design them in such a way that they're specific only to one gene. And this problem is resolved by making primers that are longer, so within the 18 to 25 base pair range. As I just mentioned, another important parameter is the primer melting temperature. And the melting temperature is the temperature at which half of the primers denatures from the DNA. So here you can see this in this graph. So you can see the temperature on the x-axis and the percentage of bases that is unpaired on the y-axis. So you can see that at a low temperature, almost no bases are unpaired. So it means all of them are paired. and the DNA is in its double-stranded form. On the other side here, at a very high temperature, we can see that almost 100% of the bases is unpaired. So this means that the DNA is denatured and is in its single-stranded form. And the melting temperature is then found here at this place where 50% of the bases is unpaired. So this means that half of the primers denatures from the DNA. And the ideal melting temperature for primers used for qPCR is between 59 and 61 degrees Celsius for both primers. The reason for this is that their melting temperature needs to be close to the annealing temperature that is used in qPCR program. And this is often around 60 degrees Celsius. So the annealing temperature is the temperature used in the second step of each qPCR cycle, where the primers will anneal to their template. And the melting temperature of primers should be close to this annealing temperature. And melting temperature is mainly determined by two factors. The first one is, as we discussed, the primer length. So generally, longer primers will have a higher melting temperature because it requires more energy to separate the primers from their template when they're longer. Separating a longer piece of DNA requires more energy. And the other factor that determines the melting temperature is the GC content of the primer. So primers with a higher GC content generally have a higher melting temperature. And the reason for this is that guanine and cytosine are coupled by 3 hydrogen bonds, whereas thymine and adenine are coupled by only 2 hydrogen bonds. And as such, denaturing guanine and cytosine requires more energy and hence results in a higher melting temperature. What would now happen if the melting temperature of our primers is too high? So this would mean that the melting temperature of the primers is much higher than the annealing temperature used in the qPCR program. Well, in that case, the primers will also bind non-specifically at the used annealing temperature. So this means that they will also bind to other locations on the cDNA where there are some base mismatches. On the other hand, when the melting temperature of the primers is too low, the primers will have already denatured at the annealing temperature of the qPCR program, so hence they will not be able to amplify the DNA. Then another important parameter to consider is the self-complementarity of the primers. So as the name already indicates, this is the likelihood that primers will bind either to themselves or to each other. And this value should always be as low as possible. And as you can see, there are different possibilities for self-complementarity. So when this value is too high, you will have an increased chance on the formation of primer dimers. And these could be either self-dimers or cross-dimers. Self-dimers would mean that 2 forward primers bind to each other or that 2 reverse primers bind to each other, whereas cross-dimers indicate a dimer consisting of 1 forward and 1 reverse primer. And in addition, high self-complementarity would also increase the chance of on hairpin formation. So this is what we discussed previously, that sequences at the 2 ends of the primer are complementary to each other and will hence bind to each other, creating this loop structure. Then a last factor to consider when designing primers is the location where they bind within the cDNA. So ideally, when you have the choice, let's say you use an online tool to design primers and you have different possible primer pairs that meet the requirements that we discussed before. So a good amplicon length, good primer length, good primer melting temperature. Then ideally, you should go for primers that bind closer to the 3' end of the cDNA. And the reasoning behind this is that mRNA is quite prone to degradation. and the degradation will mostly take place at the 5' end. So if you want to still have intact mRNA, you're more likely to find it at the 3' end of the molecule. In addition to that, another reason to pick primers that bind more towards the 3' end of the cDNA is that our cDNA synthesis to convert mRNA into cDNA is performed using oligo-DT primers, and these bind to the poly A tail, which is found at the 3' end of the mRNA. So hence, there is a higher likelihood that this 3' end of the mRNA is correctly converted into cDNA. However, remember that we also use random hexamers to prime cDNA synthesis from various locations within the mRNA. So normally, all mRNA is properly converted into cDNA, but still, the largest likelihood you will find at the 3' end. And then another reason for choosing primers more towards the 3' end of the cDNA is that alternative splicing events occur more frequently towards the 5' end of the mRNA. So when alternative splicing takes place, it's more prone to happen at this 5' site. So this means that differences between splicing variants will be found mostly on that site, whereas the 3' site will mostly be the same between splicing variants. And generally, when we want to measure the expression of a gene, we want to measure the expression of all splicing variants. Of course, it could be the case that you want to investigate specific splice variants. In that case, you should also design your primers accordingly. But in most cases, this is not our main goal, but we just want to look at the general expression of a gene, which means that we want to look at the expression of all splice variants. And that would be the easiest when you have primers binding more towards the 3' end of the cDNA. But then another important thing to consider when designing primers is their binding location with respect to junctions between two exons. So as we already discussed during the previous lecture, in qPCR, it's very important to avoid amplification of any possible residual genomic DNA, because during real-time PCR, we want to look at gene expression, so we want to have a look at the mRNA level. However, as you know, mRNA is converted into cDNA, and the primers that we design will no longer be able to distinguish between cDNA or genomic DNA. So to avoid this, of course, we perform a DNase treatment to get rid of any residual genomic DNA, but there is an extra safety that we can build in, which is to design our primers either spanning a junction between two exons or flanking a junction between two exons. And this will become more clear when you look at the figures at the bottom. So here, the first option is to design primers where one of the two primers in a pair spans an exon-exon boundary. And you can see this here. So at the top, we have the sequence of the genomic DNA, where we have a first exon, then an intron, and then a second exon. In the cDNA, as this is derived from mRNA, where the introns have been spliced out, of course, the intron is gone. The two exons will immediately touch each other. And what will happen then is if you design one of your primers such that it is on this boundary between exon 1 and exon 2, this primer will no longer be able to bind on the genomic DNA, because here, this, let's say the tail part of the primer will still be able to bind here in exon 1, but then this head part will no longer be able to bind because it's complementary to this part of the sequence here at the beginning of exon 2, but in the genomic DNA, there is an intron in between. As such, this primer will not be able to bind, and no products can be generated from genomic DNA. You will only be able to generate a product from cDNA with these primers. So this is the most ideal situation, and this is what you could go for when designing primers. However, often it's not very easy to design primers in such a way that one of them binds on top of an exon-exon boundary. And the reason is, of course, because these primers have to check all the boxes regarding the parameters that we've discussed before. So primer length, amplicon length, primer melting temperature, and also their self-complementarity. So sometimes it's very difficult to find primers that tick all of these boxes, and then also bind on top of an exon-exon junction. So therefore, what's sometimes easier is to design primers in such way that they don't bind on top of an exon-exon junction, but around a junction. So this means that they would flank the junction. So in this case, here at the bottom, we have the cDNA, where the forward primer binds an exon 1 and the reverse primer binds an exon 2. These primers will still be able to bind genomic DNA because again, the forward primer can bind on exon 1, the reverse primer can bind on exon 2. However, in cDNA, there is no intron in between, and this product from the forward until the reverse primer will be between 90 and 150 base pairs, whereas in the genomic DNA, there will be an intron in between. And in case this is a long intron, this amplicon size will be much bigger for genomic DNA than for cDNA. There will be insufficient time to generate or to amplify this product in a typical qPCR cycle, and hence no product would be formed. Sometimes, however, it's possible that this intron in between is not very long, and as such, the amplicon for genomic DNA is only slightly longer than that for cDNA, and then the product might still be formed from genomic DNA, but in that case, this product will be slightly longer, which means that it will have a higher melting temperature, and as such, you would be able to detect this on the dissociation curve, because then you would see a peak at a certain melting temperature for the cDNA product and an additional peak more towards the right at a higher melting temperature for the product formed from genomic DNA. In case you do see this does of course mean that the CQ value that you obtained for that sample is not reliable and that you should omit the sample from the data analysis. Then another thing to consider regarding the binding location of primers that you design is that they should be specific. So let's say you want to develop primers for a specific gene. In that case, the primer should only be able to bind to that gene and not to other genes in the genome of your organism of interest. And this is something that you can check using BLAST. So on NCBI, you can go to the page for blasting. You can blast the primer sequences. And what you should look at then is the E value that you obtain. So in this case, I made an example here. I designed primers for cyclin B12 gene from Arabidopsis thaliana plants. And indeed, I saw that my primers are able to bind to the specific gene that I've designed them for. And I can see that because it has a very low E value for this gene. And this is the case for both splicing variants of the gene, because you can see this here, the number 1 and number 2 behind the gene names indicate the splice variants. So you can see the E value is low, which means that the primers will bind that gene with a high specificity. And then we can see that the next genes in the list have a much higher E value. And this means that their likelihood to bind to this to these genes is much lower. So what you should check here in the BLAST output is that, first of all, the E value for the target gene, which you designed the primers for, is low. And this should be the case for all transcript variants, unless you want to specifically determine the expression of a specific transcript variant, but this will usually not be the case. And you should also check that the E value difference between your specific gene and the other genes in the list is as large as possible. And here the E value is corresponding to the number of expected hits of similar quality that could be found just by chance in a database of the same size. So this means here that a low e-value indicates that it's very unlikely that this alignment was found just by chance. So now you know which parameters to take into account when designing primers. So this is, of course, something you should not do by hand, but many tools exist that can assist you with this. A very often used one is, for example, Primer 3 Plus, but there is also other tools such as the Integrated DNA Technologies Primer Quest tool, the Eurofence Genomics qPCR Primer and Probe Design tool, SnapGene, CLC Genomics Workbench, and many others. Some of these are just online websites where you could fill in different parameters in order to design primers that meet your wishes. An example of this is Primer 3 Plus. So this is just a website that you can go to and it's free to use. You don't have to install anything. Some other softwares you might need to download onto your computer, and some of them you might have to purchase. But anyway, there is many different tools that you could use for this. So next, I will give a demonstration of how to design your own primers step by step. And then afterwards, as an assignment, you can design primers for different genes of interest.